Hello, everybody. Let's start. We have want to get through some stuff today. You guys can hear me, right?
Yes, sir.
Yes,
Great.
sir.
So let's see. One logistics business that we did say that there will be an exam. So I don't want it to be a formal exam or anything. What I'll do is midsem week is about 2 weeks, 1.5 weeks away, or 2 weeks. I'm not sure. But what I'll do is by this weekend, I will post something on Moodle, and I will post two questions, uh, and you could attempt either one of the two, and the submissions will be due last day of the midsems, and you can just email it to me. And the nature of the two questions will be such that one will be sort of more suited for people who have background in biology, and the other one for people with a more quantitative background. But you're welcome to attempt any one of the two. And again, this is going to be open-ended. There may not be even a right answer or a wrong answer, but mostly I want to study two things. One is, what is the approach that you take uh, towards coming at the possibility of a solution? And the second is, how much effort did you put in? Because especially with these type of assignments, doing something on the last day is almost a guarantee to get you a 0. All right, any questions with that? Okay, cool. So let's just continue where we left off. So any questions with Hardy-Weinberg? All right. So again, remember Hardy-Weinberg historically was a quantitative description of what is going to be the allele frequency and the genotypic frequency of three kinds of individuals if two neutral alleles were to exist at one particular locus in a population. Today, Hardy-Weinberg's context is that you know that if there is no selection acting on either of the two alleles at a locus, and all the additional assumptions that we made in its derivation, then the allele frequencies are going to be Hardy-Weinberg. Hardy-Weinberg is obviously too ideal a scenario to ever be observed in practice. And when we look at the allele frequencies in whatever system we are looking at, the deviation from Hardy-Weinberg sort of tells us that how strong are the forces which are bringing about this deviation from the equilibrium allele frequencies. So Hardy-Weinberg is sort of the baseline with which we compare everything else today. So I want to quickly wrap up this part of the section, this part of the course, and then I want to move to something called drift or effect of random effects in dictating evolutionary trajectories. So this was basically the end, the place where we left off. But imagine if selection was acting. If selection was acting, then let's say if AA genotype had fitness 1, if the heterozygous individual had a fitness 1 minus HS, and the homozygous AA had a fitness 1 minus S. H could be anything. If H is 0, then basically what we are saying is that capital A is dominant over small a. If H is 1, then we are saying that small a is dominant over capital A. If H is half, there is no dominance effect, and all the intermediate scenarios. And to begin with, before selection started acting, maybe there was an environmental change which ascribed these different fitnesses to the three genotypes. And prior to this environmental change, all three genotypes had identical fitness. But now because of this change, fitness has changed. So initially, to begin with, in our experiment, the fitness of the frequency of the three genotypes was at Hardy-Weinberg. So these were the three fitnesses. But now because fitness, I'm sorry, these are the three frequencies. But now because fitness has changed, in the next generation, this is going to be just p square. So we're just saying frequency times fitness. This is just so for each one of the three in the next generation, the number of individuals can simply be frequency times fitness. So you get these three frequencies in the next generation, and when you sum up, this is some sort of a measure of the total number of individuals in the next generation. I mean, this is pretty straightforward, but we are going to get into some quantitative aspects of the course now. So it would be useful if you were to just try these things by your hand once. It's pretty straightforward. I mean, so the addition of these three comes out to this particular expression, because when you add, you get p square, plus when this multiplies with this, you get q square, and when you have 2pq multiplied by 1, you have 2pq. So these three terms, p square plus q square plus 2pq, we know this equals to 1. And the remaining two terms are just the ones that are here as the second term in the sum. So if this is the total number of individuals, then the total number of alleles which are carried at this particular locus is just 2 times this number, because every individual is carrying 2 copies. So that's the total number of alleles. And if we are interested in frequency of allele A in the next generation, then that's just equal to 2 times this frequency, because all these p square individuals are carrying 2 alleles of capital A, and all these individuals are carrying 1 allele of capital A. So it will be 2 times p square plus 1 times this particular expression, which gives me this. So the frequency of allele A in the next generation changes from whatever the frequency was here. Well, basically it was p plus q. The allele frequency changes to this. And now the question before us is that what is going to be the shape of this curve for these two scenarios? What do you guys think? Oops. So what I'm going to track now is this methodology gives me a way to calculate the allele frequency of any one of the two alleles as I go from one generation to the other. If I keep applying this over and over again, I will keep getting the frequency of the capital A allele in the subsequent generations. So if I apply, if I do this type of an analysis for a long enough time, I can track how is the frequency of allele A changing as number of generations increases. So tell me, what do you think the graph's going to look like for the case h equal to, let's do h equal to 0 first. So h equals 0 basically means that capital A is dominant over small a. What do you think the graph is going to look like? And let's say this is the starting frequency. To begin with, individuals carrying allele A were very, very few. So allele frequency of A is very small. H is 0. What do you think? Anybody? What is the challenge in sort of attempting this?
So can we see the equation again?
Yeah. Well, I don't want you to think in the context of equation, but I want you to think in terms of logic. H is 0.
Yeah.
Don't worry about the equation. The equation is only there. Yeah, if that is an issue, I don't want you to think about it in the terms of the equation, because I mean, if you're trying to plug in numbers and all, I don't want that. Because that's only for tracing the exact curve. That's the only purpose of the equation. For the purpose of coming up with a sketch for this graph, I want you to think logically.
So if capital A is dominant, we will see a slow rise initially, but
Uh,
eventually the whole population will be taken over by the dominant allele.
okay, why do you say
So
that so something like a sigmoidal curve, do you mean that?
yeah, kind of sigmoid. Yeah.
Okay. So why do you say that it will be slow initially?
Um, because initially the dominant individuals,
Hmm.
individuals with dominant genotype
Hmm.
will have less less proportion of the population which
Correct.
can give the dominant ones.
Correct. So,
So as the that proportion will increase uh, slowly. That's why we will have a sigmoid kind of.
okay. Anybody else? Is the question clear to everybody? Is the question not clear to anybody?
Yeah, it's clear. Just
Okay.
one point key. A won't completely take over
Okay. Why do you say that?
because there is no reason for the heterozygous to be selected against.
Hmm. Yes. Yeah. So that's a good observation. So what Akshat is saying is that this will this will increase in frequency. We don't know what is the precise trajectory, but there is no reason for us to believe that A will reach a frequency 1, because when you have the entire population, let's assume that entire population is capital A small a, entire population is heterozygous. You couldn't increase an individual's fitness anymore, right? uh, Everybody is at fitness 1 and we couldn't do any better. Is there a problem with that, Akshat?
Problem with key. I didn't get the problem.
The Well, so the issue is this, that a population which is comprised entirely of heterozygous individuals, right, is going to produce 25% small a homozygous individuals, correct?
Huh?
And that's going to mean that many of the offsprings are not as fit,
Yes.
right? Compare that with the population where everybody is a homozygous individual, capital A, capital A. The offsprings of this population are going to be much fitter,
Yes.
right? And that would mean that the small a allele is driven to very, very small frequencies in the population, because you can't have too many heterozygous individuals in the population, because then you're going to have a sizable fraction which is small a,
Yeah.
right? This particular cost of fitness, so essentially what we are saying is that if you have too many heterozygous individuals, then it doesn't matter as far as fitness of the present generation is concerned, but it does matter in terms of what is going to be the genotype of the individuals in the next generation, right? That particular cost in fitness, that if the population is entirely heterozygous individuals, its fitness is small because it's going to produce small a homozygous individuals. This particular cost of fitness, which is associated with what are you going to produce in the next generation, is called segregation load. Does that make sense to everybody? What is segregation load?
Can you repeat that, sir?
Yeah. So Imagine a population where everybody is heterozygous, everybody is capital A, small a. And there is another population which is capital A, capital A. Now, in both these populations, H is 0. In both these populations, every individual is at fitness 1, right? So there is no difference in terms of how fit the participating individuals in both these populations are. When mating takes place in this population to produce offsprings in the next generation, then everybody is going to be of fitness 1 because you only have homozygous capital A individuals, right? However, when mating takes place in this particular population where everybody is capital A, small a, then because of random mating, 25% of the individuals are going to be a genotype small a. And these 25% of the population are going to be of fitness 1 minus S, which is obviously lesser than the fitness of the parent population capital A, small a, right? Hence, if you have a heterozygous population, then, and even though there is no dominance, H is equal to 0, I mean, there is dominance and H is equal to 0, then even though the parent population, everybody was at a fitness S, a fraction of your population in the next generation is going to be lower fitness just because you are producing the individuals with homozygous recessive allele, which is small a here. And this resultant decrease in fitness is called segregation load. Because this genotype, this particular individual with this genotype had a fitness 1 because in addition to small a, it also had this capital A allele which made up for the fact that it was carrying small a. Now these two got segregated. So I am a small a capital A and I mate with someone who is also small a capital A, and our alleles got segregated because of meiosis, and hence the offspring that we produce is of a genotype small a. And because of small a's segregation, it got separated with the allele capital A, the fitness of the offspring is going to be 1 minus S. Hence there is a fitness cost that the offspring paid. And this cost was paid because of segregation of the two alleles that both the parents were carrying in this particular case. Such a cost of fitness, such a loss of fitness is called segregation load. Does that make sense?
Yes, sir. But while we are talking about the fitness, we will be seeing at the fitness of the population, right?
Correct. So mean fitness is just going to be a function of that, right? In a capital A
Yeah.
population, you're not producing, everybody has fitness 1, so mean fitness is also 1. But in a small a capital A, you're producing a bunch of small a's, and hence mean fitness decreases, right?
Yeah.
Cool. So let's go back to our graph. What do you think about our graph? What is going to be the trajectory of this curve? And I would like to, us to develop some sort of an intuition about this kind of a phenomena. H is equal to 0. We are not looking at equation.
The rate of increase will slow down with number of generations.
Why is that?
Because let me phrase it.
Okay, while you think, others, I'm going to start picking on people. Are you guys sleepy today? I take that as a yes. I am going to start picking on people now. Gaurav, what do you think? Gaurav is not sleepy. In fact, he is asleep. So let that be. All right, let me ask you a question. This allele frequency is really small, okay? So for all practical purposes, let's assume that, I mean, there is one individual in thousands, which is of the allele capital A small a, right? So if I were to ask you that at this t equal to 0, what is the mean fitness of the population? What would you say?
So 1 minus S.
Anirudh?
Could you repeat that again, please?
Yeah. So the question is that at this particular instant, when an experiment started, let's imagine there was a large population and there is one individual who's carrying one copy of capital A allele because a mutation just happened, right? So we have one heterozygous individuals, everybody is homozygous for the recessive allele. At this particular instant, at time t equal to 0, what is the mean fitness of the population is the question. And yes, you're right.
Yeah, it would be 1 minus S in this case.
Yes, exactly. So fitness is 1 minus S at this particular instant. What is the fitness of this one heterozygous individual at this instant?
1.
It's 1, right? So what is the relative advantage that this one heterozygous individual enjoys over the mean fitness?
It'll be S.
Will be S, yes, right? So the individual's fitness is 1, and the difference between the two fitnesses is S. So the relative fitness is S. Exactly, perfect. So in other words, what we mean to say is that the fitness of this particular individual is S units more than the mean fitness of the population. Does everybody agree with that statement?
Yes, sir.
Now let's imagine a future scenario where allele frequency of A has increased to some value, right? We don't really care for the numerical value. Maybe the allele frequency is somewhere over here, right? What do you think has happened to the relative advantage of the heterozygous individual when the allele frequency of A has increased to some number? Let's say it was 0.2 allele frequency of capital A. What has happened to the relative advantage of the head of any heterozygous individual over the mean fitness of the population? Remember, at the beginning of the experiment, this relative advantage was S. What has happened to that fitness? What has happened to that advantage now?
It has decreased.
Why has it decreased?
Because mean increases as mean increases, the difference decreases there.
Absolutely. Absolutely. So what Namrata is saying is exactly right. That as we move forward in time and there is a greater fraction of individuals carrying capital A allele, we have more heterozygous and we have more homozygous individuals which are carrying the dominant allele. Some individuals will be capital A, some will be capital A, small a. All these individuals are at fitness 1. And because there are an appreciable number of these individuals in the population now, the mean fitness of the population is no longer 1 minus S. The mean fitness is now more than 1 minus S, but it's obviously less than 1. What that means is that the relative advantage that heterozygous and capital A homozygous individuals, the relative advantage that these guys enjoy over the mean population's mean fitness is now smaller than S. Does that make sense?
Yes, sir.
Yes.
Yeah. So and I mean the math is, yeah, I wrote down the math so that you can verify. If you were to recursively keep plugging these guys in, you will see the trend. But intuitively, this is how I want you to think about it. That because as we move forward in time, the relative advantage that I as a heterozygous or a capital A individual enjoy over my relative advantage over the population's mean fitness is decreasing, and hence the alleles that I am carrying are going to increase in frequency with increasingly smaller speed as I move forward in time. The rate at which an allele penetrates into the population and spreads in a population is proportional to the relative advantage that the individual carrying that allele has over the population's mean fitness. And this relative advantage keeps on decreasing as we move forward in time. And hence, in this particular case, when h is equal to 0, the rate at which allele frequency of A is increasing continuously decreases as we move forward in time. So the first graph that we are looking at, h equal to 0, is going to look something like this. That initially it's going to start very rapidly because the relative advantage is s. But as that s starts to decrease, the rate of increase starts to decrease, and it will asymptotically approach 1. Does that make sense to everybody intuitively?
Yes, sir.
Yeah, Anirudh, go ahead.
This was also seen in the case of the diminishing epistasis theory as well, right? But it's a bit different in that case, but I'm just asking.
Well, if you're saying that, does the nature of the plots look alike? It
Yeah,
does.
yeah.
It does. Yeah.
But there
Uh,
they, I'm
but
getting a bit
the contexts are very different.
so that's how a beneficial mutation would affect the general fitness, and this is just how one allele's frequency changes over time. That's
Yeah.
the difference.
So let me answer and let me know if you know that is a satisfactory explanation of some degree. This is a case where we are talking of a population. And in a population where there was no genetic variance, one individual carries a mutation, and we are interested in how that beneficial allele is spreading through the population, right? So x-axis is time. Does that make sense?
Yeah.
The other case, the diminishing returns epistasis, what you're saying, that you're not looking at a population there, you're looking at an individual. And what you're asking is that if you now the x-axis in that case is number of beneficial mutations I'm carrying, right? So in this individual, in one particular individual, if you were to introduce one beneficial mutation, it would give me a benefit of, say, s units. But now while I'm carrying that one beneficial mutation, if you were to introduce another one, the resultant benefit of the second one will be less than s, and then further less and further less, and so on and so forth. So that is a phenomena that's seen because of epistasis that exists within a genome. This is just how an allele spreads in a population. There is no epistasis here, right? This is only one locus, and we are seeing how that allele is spreading in a population.
Right. Yeah.
Right? Yeah.
Yeah.
Cool. Any other questions? All right. So remember, allele, Akshay, does that, I guess I answered whatever you were trying to phrase, but anything you want to add?
Yeah, perfectly.
Cool. So remember, allele frequency increases, and its frequency and the rate of increase in rate of increase of allele frequency is proportional to the relative advantage over the population's mean fitness. That's an important quantity. Let me now come to the other one, and I want you to think about this for a minute or so. What happens when H is equal to 1? Same scenario, everything's the same. And we saw that in H equal to 0, again, you can work out the math yourselves. This is just a simple Excel exercise. But intuitively, we saw that this would have a trajectory like this. What happens? What do you think the graph's going to look like when H is equal to 0? I'm sorry, H is equal to 1. Think aloud.
So there'll be a small blip in the graph, but eventually it will die out.
Blip matlab?
As in, there will be a small population which will emerge having the heterozygous
Yes.
genotype,
Yes.
but due to the relative fitness being lower,
It's
they
not
will
lower,
die.
right? It's not lower. No, first complete whatever you're saying.
So when H is equal to 1, we are saying that,
So let me make sure we are on
okay,
the
right,
same page. Everybody
Hmm.
AA, everybody is small a, small a. H is equal to 1, right?
Yeah,
And in this scenario, we have a mutation where small a comes up, but it's the homozygous capital A which enjoys a fitness advantage and not the heterozygous genotype, right?
right.
So what do you
So
guys think?
basically the heterozygous will have same as the homozygous recessive.
Yes.
So this graph will be like what I suggested earlier,
Hmm.
a sigmoid,
Why would it be a sigmoid?
maybe. Um, like the homozygous capital A still has fitness equal to 1, right?
Yeah, yes.
So we will see that the population of heterozygous will grow, and as it is growing,
Hmm.
the population of homozygous dominant will also grow,
Hmm.
and by considering random mating,
Hmm.
this population size will keep on increasing.
Got it. Yeah. Anirudh, you wanted to add something?
No, no.
All right. What Yasha said is just perfect. In this scenario, what's going to happen is that you have this one individual. Now, this one individual is going to make a few progenies. Everybody is making a few progenies, right? And then the allele frequency of capital A has gone up. But because this individual has mated with another homozygous small a individual, because there is no other heterozygous individual in the population, this guy mates with a small a, or more than one small a individual and produces a few progenies. And then basically it takes a long time. Even now, so for a long time, what's going to happen is that the number of that the capital A allele is only present in the heterozygous individuals. It's not present. You are not producing this homozygous individual because there are so few heterozygous individuals in the population. The chances of them mating are extremely small. And remember that even if two of them mate, there is only a 25% chance that you're going to produce a capital A homozygous individual. So, and before this happens, basically there is no selection acting because all individuals are of the same fitness. So there is no selective advantage that this enjoys. So the allele frequency of capital A will keep itself low for a long time. And then exactly as Yash said, that random chance will ensure that at some point some capital A individuals are going to get produced, and as soon as that happens, you have a fitness differential that kicks into the system, and as the number of capital A individuals increases, then this fitness differential comes into picture, and the resulting graph that you see is of a nature which is almost flat to begin with. There's a long waiting time, and then as capital A individuals come into being, it rapidly rises and then approaches 1 again. Does that make sense to everybody? At some intuitive level? Okay, cool. So, yeah, so basically just what we discussed. I think I have these the other way around. I think I was looking at the dominance relationships the other way. H is equal to 0 is capital A dominant. Yeah. So these should be the other way around. This is the graph for H is equal to 0. This is the graph for H equal to 1. And all of this comes from the fact, comes from mathematically, all of this comes from this particular relationship. And you can try this in Excel or something. And the trajectories would come out to be the same. There is a special case when H is negative. Okay. And in that case, when H is less than 0, what you're saying is the heterozygous individual is actually carrying the advantage over the rest of the population. Both kinds of homozygous individuals are less fit. Heterozygous individuals are fitter than everybody. And again, if you do the math here, the trajectory of that particular scenario is going to be something like this, that A will increase in frequency, but then it won't go to frequency 1 because these guys are less fit. It will just saturate somewhere in the middle, and that's basically it. It stays there because either of the two, eliminating A and A going to frequency 1, decreases the fitness of the population. So in that case, again, this relationship will tell you the value that it saturates it. The precise value of that is obviously going to depend on how much was S and how much was S, how much was H and how much was S. But basically, it doesn't reach frequency 1 or 0, stays in the middle. Can anybody give an example where H might be negative? Rather than thinking of H being negative, think of a scenario where a heterozygous individual enjoys an advantage over both kinds of homozygous individuals.
Sickle cell anemia.
Yeah, perfect. Sickle cell anemia is the most well studied, right? So you have a mutation in hemoglobin, and as a result of that, your oxygen carrying capacity is decreased, is compromised. Now, where is that advantage enjoyed? You have to be in a particular type of a location for the heterozygous individual to enjoy that advantage.
Malaria prone regions.
Malaria prone areas.
Exactly. Right. So if you don't have that mutation, then you are susceptible to malaria. But so I mean, all of this is only relevant if you live in a malaria prone area. If you're living in Western Europe or something, there is no benefit in carrying that mutation. But if you're a homozygous individual carrying both the mutations, both the alleles are the mutant versions, then your oxygen carrying capacity is compromised and you suffer because of that. If you're homozygous for two healthy alleles, then your oxygen carrying capacity is not compromised, but you're susceptible to malaria. But if you're heterozygous at that particular locus, then your oxygen carrying capacity is not compromised. So the healthy allele is dominant over the mutant allele, and your susceptibility to malaria goes away. And as a result of that, in regions of Africa where malaria is widely present, what you see is that mutant allele is not eliminated from the population, it stays there. Interestingly, whenever you have H less than 0, what that means is that a bunch of your, what that means is that the fittest individuals in the population are going to be the heterozygous individuals. But we just discussed that having heterozygous individuals in a population as the fittest individuals has a cost associated with it. And in this scenario also, if I am a heterozygous individual in Africa and I am fit because I'm carrying one copy of each allele, and if I mate with someone who is also carrying one copy of each allele, then there is a 50% chance that we are going to produce an offspring who is homozygous at that allele, and the homozygous individuals we know have a fitness cost in such regions. And this is another example of what we discussed earlier, segregation load. This happens when the heterozygous is at least as fit, if not more fit, than both the homozygous genotypes at a particular locus. Does that make sense?
Yes, sir.
Okay, all right. So what we discussed basically was just one allele at one particular locus, but the dynamics of how allele frequencies change in a population are obviously much complex. Not only do you have many loci, and at each loci many alleles, you have recombination which creates new alleles. You have recombination which links and delinks two alleles which are next to each other, and so on and so forth. So the actual evolutionary dynamics in a sexually reproducing population is going to be much complex than what the overtly simplistic scenario that we have just discussed. We will, and we can play with it. We can make up scenarios where, you know, on two different chromosomes there are two different loci. These two loci have two different alleles, and each one has a dominance relationship within itself, and what are going to be the dynamics of that. But that's basically just building on the complexity of what we just discussed, and we can take that complexity to any nth degree. So I'm not going to spend too much time talking about that. Instead, what I do want to talk about is the second major force which brings about allele frequency, which brings about change in allele frequency, and that is the action of drift or randomness in an evolutionary process. All right. So let me ask you a question first. That imagine a population. Actually, let's do it the other way around. Imagine, so this is a popular game which is called, well, it's not really a game, but think of it's called marbles in a jar scenario. Imagine a jar which contains n marbles. Each of the n marbles has a different color or a design associated with it. So you have n marbles, each with a different color or design. Just let me know if I don't make sense at any point. So the frequency of each design in the population, since every marble is unique, is simply 1 upon n. What we are going to do is that we put our hand in this jar and pick out one marble randomly from it without looking. And let's say that that marble is this particular design, right? So we take it out, we take a look at it. And then after we take a look at it, we do two things. We put the selected marble back in the jar. So I drew this particular marble from this jar and I put it back in now. So my original jar again contains n different marbles because I put the one that I drew out back in that jar. And I take a second jar and I add a marble of identical design in this new jar, right? So because I drew this one, I put another marble of an identical design in the next jar. And I keep on doing this till jar number 2 contains exactly n balls or marbles, whatever. So, I mean, because I put this particular design back in the jar, there is a finite chance that I'm going to, in the subsequent n-1 draws, I'm going to draw this particular marble again. And as a result of that, in the second jar, I might have more than one ball of this particular design or any other design. I mean, this might get picked twice or more in the subsequent n-1 draws, and so on and so forth. That's the rule of this quote unquote game that we're going to be playing. So what is the frequency? If we ask ourselves that what is the frequency of each design in transitioning from jar 1, where everybody's frequency was 1 upon n, in transitioning from this jar 1 to jar 2, there might be some designs which never get picked, and frequency of those designs is going to be equal to 0. Some of the designs will remain at the same frequency, and that frequency is just 1 upon n. And some, perhaps like this one, which gets picked more than once, the frequency in jar 2 is going to be greater than 1 by n. Right? The question that I want to ask you guys first is, why did the frequency change? Because every ball had a finite chance of being drawn, and that finite chance was 1 upon n for every single ball, right? So the first question is, why did the frequency change? And the second question is, that what is the ultimate fate of this marbles in a jar game? What happens if I keep on propagating this jar, one jar after another, and I do it for a long enough time? What is the quote unquote ultimate fate of this marbles in a jar game? Tell me. What do you guys think? Is the scenario clear? Is what we are discussing clear?
Yes, sir.
And
So the frequency probably changed because it's random picking.
Exactly.
And eventually there's chance that only one marble will go in the other jars as well.
Only one marble, or do you
Yeah,
mean to
because
say one design?
one design.
Yeah, exactly. So eventually, you're right on both the counts. What's going to happen is, why is frequency changing, right? Frequency is changing because this is a process which has an inherent randomness associated with it. Because of that randomness, one ball might get picked more than once, and hence that's overrepresented in jar 2. Whereas it might be some balls might never get picked, and those balls are going to be eliminated from the population. That's one. And secondly, that the eventual fate of this game is, there is only one fate, that eventually you will only have one particular design that populates the entire chart. And again, that's going to happen because of chance events, This is important as far as evolutionary dynamics is concerned. That because if we were to play this game hundreds and thousands of time and take an average, the average will show the average of all these. What will the average show? If I play this game, let's say with 10 marbles, different designs, and I play this game 10,000 times, and if I take an average, what will the average trajectory look like? Is the question clear? Is the question not clear?
So could you repeat the question?
Yeah. The question is that okay, let me take a step back and show you a way to represent this. And this is, I mean, this is marbles in a jar, but you see this type of a plot in many papers these days. This is jar number. So this is jar 1, jar 2, and so on and so forth. This is number of balls, 0. This is 1, 2, and so on and so forth, going all the way to n. Right? Design number 1, its frequency was this, 0 to 1. This was design number 1. Design number 2 was this 1 to 2. So these little 2 bars, the distance between these two was design number 2. This was design number 3, design number 4, so on and so forth, design number n-1, and design number n at the end of it. Does that make sense? Is the representation clear?
Yes, sir.
Yes,
Yes, sir.
no, okay, all right.
Yes, sir.
As we move from the first jar to the second jar, see what has happened. Basically, the frequency of this first design, so this first design never got picked in jar 2. As a result of that, basically this, I mean, how do I explain this? This section here, the width of this section represents how many balls of design 1 are there. So in jar number 1, there is 1 ball which has design number 1. As I move to jar 2, design number 1 never got picked, and hence this particular area, this particular segment shrunk to 0. So there is no ball of design number 1 in the second jar. If you look at this second segment, which represents design number 2, this widens, right? This means the relative segment length has increased from this much to this much as we come to jar number 2. So maybe this particular ball got picked 3 times. Look at this particular one. This was 1 ball, which was design number 3. But as we move to the second jar, this segment shrunk to 0, which means design number 3 also got eliminated. Again, design number 4, its width is increasing. This was 1 ball, but maybe by the time it got here, it was 2 balls, and so on and so forth. And similarly, for each design, as we move forward, at any particular jar number, if we look at the segment, the length, if we look at this length of this vertical segment in that particular section, we can tell the frequency of that particular design at any particular jar. Does this graph make sense now?
Yes, sir.
Yes, sir.
Right? Anybody to whom it doesn't make sense? Cool. So what is happening in this particular case, for instance, that this first design went extinct immediately. This second design went extinct immediately. This third design, this also went extinct. The second design it sort of hovered around. It increased and then decreased in frequency. Then there was an increase in frequency, because remember, for the second design, we are counting this entire segment. Design number 1 doesn't matter now because it went extinct right here. And then it hovered around, and eventually it went extinct too at pth jar. By the time we get to this pth jar, all the other designs also got, if you look at any one particular trajectory, all of them went extinct. And it was actually this design number 4 which basically took over the entire population in the jar, and now every marble in the jar was design number 4 by the time it comes to this particular jar number, right? So this is a scenario of one particular realization of this experiment, right? Whenever you have a stochastic event, one play of such a stochastic event is called a realization. So this is an example of one such realization. Does this make sense to everybody? Because this is a graph that's going to come many times in many of the subsequent readings that we're going to do in the semester. Make sense?
Yes, sir.
All right, cool. So now let me ask you another question. Now let me go back to the question that suppose I did this experiment 1,000,000 times, right? Every such iteration, and let's say n is equal to 10. There are 10 balls to begin the experiment. In every iteration of this, in every iteration of this marbles in a jar game, I'm going to get a different trajectory, right? And I plot each one of those. So I have 1,000,000 such graphs. And if I take the average of all such 1,000,000 graphs, what would the average trajectory look like? Come on, guys.
So is it a straight line, I mean parallel line to x-axis?
Yeah, absolutely. Exactly. What's going to happen is that when I do this experiment 1,000,000 times, because the number of times I'm doing this experiment is much more than the number of participants in this game, what's going to happen is that sometimes this design number n-1 will win, sometimes design number 1 will win. There is absolutely no reason for one design to win over the other. All of this is happening because of random chance. And if I do this 1,000,000 times and there are only 10 different designs, then roughly speaking, each one of the designs will win 100,000 times. So, and if I average all of them, basically what's going to happen is that sometimes this will go extinct at first one, sometimes this will increase in frequency, sometimes it will stay the same. When I average this over 1,000,000 different trials, the trajectories are roughly going to stay the same, stay the same exactly as what we started with this in this particular experiment. So the averaged out trajectory over 1,000,000 runs of this is just going to be straight lines, and as if frequencies do not change with time. Does that make sense to everybody? Yes, no?
Yes.
Others?
Can you explain it once?
Yeah. So what is happening here is that in this particular scenario, design number 4 won, right? But if I do this another time, there is absolutely it's not as if design number 4 enjoys some sort of an advantage over the other designs. design number 4 was as likely to win as design number n-2. So in another realization of the same experiment, if I started from scratch again, design number 1 might win, design number n might win, anybody might win, right? All of them have an equal chance of winning, and by winning we mean occupying the entire jar with the same design. So anybody might win. So if I do the, if n is equal to 10, if I'm doing this experiment starting with 10 different designs, if I'm doing this a million times, then roughly speaking, each one of the designs will win 100,000 times, right? So which means that when I come to this particular point, out of a million, 100,000, if I come to this point, out of a million times, 100,000 times design number 1 won. So 100,000 times design number 2 was the winning design. So if I do the average and plot frequencies, this is a frequency plot for one particular realization. If I plot the average of the frequency plots of a million trials, then basically, because no one design enjoys advantage over any other design, I'm just going to get straight lines. That the frequency, it will look as if the frequency always remains the same. All right, does that make sense?
Yes, sir.
Okay. This now poses a problem when it comes to studying evolution. First of all, does anybody want to take a guess of what is the, how can we link this with evolution? How can I extend the marbles in a jar game to a study in evolution? All right.
The evolution somehow.
Go ahead, Daniel.
Yeah, during a chance event separated, I guess, two populations.
Okay.
And in a particular environment, there only exists now one population, and this keeps happening, I guess. But it's a one-time event.
Yeah, yeah. So this was, yeah, sure. And there are other ways also that it can play out, and we'll look at them in just a second. But the issue is this, that now, so far, if I'm thinking deterministically, imagine the scenario of h equal to 1 that we looked at in the previous case, where the small a was the dominant one, right? When you have a capital A individual, heterozygous individuals show up in the population, that particular individual is only 1 in number. Now there is a chance that that individual, might not produce offsprings, might get run over by a train. I'm making stuff up. The point is that chance events are not taken into account when we are looking at averages. Averages would tell us that this stays horizontal throughout the time. The issue here is that populations do not behave as averages. In a realization of an evolutionary experiment in any environmental niche that you are doing out, you are only going to have one particular realization of the experiment and not average of a million particular trials. What this does, it makes things difficult for us to understand, because what this tells us is that averages won't help us. But if we look at individual realizations, there are so many individual realizations, and keeping track of all of them is going to be hard, right? And all of this is happening just because there is an inherent randomness associated with evolutionary processes. So let me just talk about what is the connect with. How can we think of an evolutionary process in a manner which is consistent with marbles in a jar game? All right. So this is just the same thing, that at the pth jar, n-1 designs are not found, they've been eliminated, and one of the designs is carried by all n. I write individuals, but balls is what I mean. What is the parallel with evolution? How many of you do not know what is a chemostat? All right. Okay, quite a few. So a chemostat, well, some of you are, how many of you don't know what is a CSTR? Okay, fine. So imagine a reactor like this. Imagine a reactor like this, which has a liquid of volume V, capital V, right? And it has bacteria growing in it. Eventually, forget about the inlet and the outlet streams. If I let this bacteria grow in this volume, what's going to happen is that it's going to use up all the nutrients that are provided here, and eventually there is going to be no more nutrients left for growth to take place. And hence growth will stall and there will be no more division. And then because of starvation and starvation induced stresses, we'll have deaths taking place here. Right? And if you think about it, growth in such an, I mean, this would be a growth experiment that you do in a test tube. Again, please make sure that I'm talking of a scenario where you can forget about the inlet and the outlet. Growth in a test tube takes place in a certain fashion, that you take a test tube, you add a bit of resources to it, and you induce it with a few bacteria. And then you let the culture grow overnight. And when you come in the following day, you look at that, you see that the entire culture now is turbid because growth has taken place. But the turbidity is not going to increase because now there is no more resources left for bacteria to grow anymore. But an important point to realize here is that during the phase that growth took place in the night, the conditions in the test tube were changing with every instant. When you started the culture, we say inoculated the culture. When you started the culture, the liquid that you started had glucose of 10 grams per liter concentration. However, 2 hours after that, it was only 8 grams per liter. And 2 hours after that, it was only 6 grams per liter, and so on and so forth. And the same is true for every other metabolite or resource that you added in that tube. That growth in a test tube environment is not a constant environment. That environment is constantly changing. Right? And obviously, when you reach saturation, all the resources are gone. And in fact, you also have the waste products that have been secreted by the bacteria into the test tube environment. So the chemistry of the environment is constantly changing. And hence, studying growth in a test tube is tricky because you're not studying the response of a population to a constant environment, but you're actually studying the response of the population to an environment which is changing every few minutes if the organisms are multiplying rapidly. To counter that, you have something like this, which is called a chemostat, or in chemical engineering, we call it a continuous stirred tank reactor, CSTR. But you don't need to know CSTRs. In this particular scenario, exactly the same thing is happening, that you have a volume capital V and you have growth taking place here. But you know that this will saturate soon. Hence, what you do is you are adding fresh media at a rate V naught volume per unit time. Right? So this could be 10-liter tank and you are adding 1 liter every hour. You control this so that it's continuously liquid is being added, fresh liquid with no bacteria. So this is just nutrients that you are added, nutrient liquid media that you have prepared without any bacteria. So this is basically just you giving food to the bacteria so that they keep on dividing. So this is 10 liters. This is being added at 1 liter per hour. But then you can't have addition for infinite time. So you're also removing liquid at the same rate. You're also removing liquid from the tank at the rate 1 liter per hour. The difference being that what you're adding is fresh media. This has no bacteria. Basically, this is just food for the existing bacteria in here. And what you're removing is actually what is present in the reactor. Usually there is a stirrer here, so there are no spatial heterogeneities. All of this is well mixed and the conditions are identical. Now this exit stream is going to contain a bunch of things. It is going to contain some of this fresh media also. Some of the constituents are going to go unutilized. It will also contain the bacteria that are present here, so those go as well. It will also contain some of the metabolites that have been secreted, some of the waste products like E. coli makes acetate and secretes. So this media will have some acetate, some of that will go, and so on and so forth. Eventually, if you let this thing run long enough, an equilibrium will be established such that the rate of removal of everything, rate of removal of bacteria, rate of removal of toxic products, is exactly matched by the rate of production of all of those things here. So if you're removing 10 to the power 8 bacteria per hour in this stream, you're also producing 10 to the power 8 bacteria in here. An equilibrium between the two is established, and hence the conditions inside this tank no longer change with time. This particular setup is called a chemostat. Does that make sense to everybody?
Yes, sir.
Yes, sir.
Yes, sir.
Cool. All right. That's a chemostat. So a chemostat is with n at a particular all right. Now let's imagine that in this chemostat, well, first of all, the connect with ecology is that you can think of almost all environments. Think of any ecological niche, a lake, a pond, soil sample, anything. Almost all environments can be thought of as chemostats. No environment can be contained within itself because it doesn't have infinite resources. There has to be an inlet stream of resources, and not just resources. It could bring in pathogens. It could bring in all sorts of things. So there has to, the point being that I'm not in any way saying that this experiment is representative of ecology. I'm just saying that this is representative to the extent that the basic principle holds, that there is a certain inlet stream, there has to be a certain outlet stream to every ecological niche that you are looking at. So with that gross oversimplification, let's imagine that this represents a niche of some sort. New resources are coming in at a certain rate, existing resources are being washed away at a certain rate, and you have a steady state in this environment. Now let us imagine that this contains 10 to the power 10 bacteria in this entire tank. One of the bacteria picks up a mutation which is beneficial to it. What is going to be the fate of this population if I let it run for 3 more days? So I have 10 to the power 10 bacteria which are genetically identical with each other, have the same fitness, but I have one individual which is a variant which enjoys a selection coefficient of S over everybody else in the population, right? And now I let this experiment go on for 3 more days, and let's assume no more new mutations come up. The only dynamics we are interested in at this point is between this one variant and the existing 10 power 10 minus 1 individuals. What is going to be the outcome of this chemostat experiment after 3 days? Is the experiment clear?
Yes, sir.
Yeah. So what is the outcome, or what are the possible outcomes of this experiment after 3 days? Yeah, Anirudh, go ahead.
So one of the outcomes would be that uh, that particular mutation could be beneficial and the bacteria divides and it grows out to be um, higher in the mutation would spread through the population.
Yeah, yeah,
And
correct. So we are starting with the assumption that the beneficial was in fact that the mutation was in fact beneficial. Yes.
the second would be in the case when it was a beneficial mutation, but by randomness it got removed from that particular tank.
Mm-hmm.
In that case, it's as though no mutation was present there.
Exactly. Exactly. So so these are the two possible scenarios in the limited context of no new mutations allowed that could happen in a setting like this. The first one, I'll just repeat what Anirudh said. First one is that since this is a beneficial mutation, it is going to spread through the population and start outcompeting the existing individuals, and its frequency will increase. So when I come back 3 days later, majority of the individuals might be the ones which are carrying this beneficial allele. On the other hand, because of the chemostat setup that I have, and I know that bacteria are being continuously removed from the population, a chance event might decide that this one beneficial individual actually was eliminated from the population before it had a chance to outcompete, start sort of spreading into the population. And if that happens, then basically what happened is that this allele is lost from the population and the population remains the existing genotype. And despite being a beneficial mutation, that particular allele never got a chance to spread through the population. And the cause of this particular effect is just that this happened because of a random chance event, right? Just as it did in a marbles in a jar game, that particular design never got picked uh, as we transition from one marble to another, right? So in a deterministic setting, if we didn't take this chance event into account, if there was no possibility of this loss of an allele take place, then the probability that this beneficial mutation is going to spread through the population, this probability was going to be equal to 1, right? However, now because of this chance event, because of this discussion on the chance event, that probability we know is going to be less than 1. Does that make sense?
Yes, sir.
Right? And this is the action of drift that we are going to be looking at next in the course. All right. So again, that's what I wrote. And obviously, let's so a different manifestation of that, and this brings it closer to, and we'll stop with that. A different manifestation of the same thing will be that in this population, every individual is carrying a unique allele at a one particular locus. However, this locus is selectively neutral, which means that it doesn't matter which allele you are carrying at this particular locus in the genome, your fitness doesn't change. So all 10 par 10 individuals are carrying a different locus, but then the fitness of each of the 10 par 10 individuals is exactly the same, which is just equal to 1. At all other loci, the genomes are identical with each other. No new mutations occur. We just add this constraint to make life easier now, at least for now. And we ask that what is the fate of this population after a long time. So given all of this, what is the fate, what is going to happen after a long time? Pavitra,
Yes, sir.
what do you think?
Can you repeat the last part?
Yeah. The What do you mean by the last part? Do you mean the entire thing?
No, 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 no. From
Hmm.
after the part that the other genomes are identical.
Yeah, so no new mutations occur. So existing variation is all we have. No new mutations occur. What is going to be the fate of this population after a long time?
Oh, new species would could form.
No, how could new species form, right? This is bacteria and no new mutations are allowed. I asked you this because the answer to this is the same as what you said about the marbles in a jar game.
Yes, sir. But then, yeah, okay, fine. Yeah, yeah.
So what happens?
No, either it survives or the mutation just survives or uh, it just goes away.
Okay. Anybody else?
So the frequency curve might look like the marble one, like with
Mhm.
different scenario, like different. Because of randomness, it might vary. The frequencies might vary.
Yeah, exactly. So now remember, every individual is carrying a unique allele. So the frequency of every genotype and genotype everybody else everywhere else is identical. The only difference in the genotype is at this particular locus. So frequency of every allele is just simply equal to 1 by n. And it's not hard to imagine that as soon as the experiment starts, we're going to lose a whole bunch of alleles straight away. And we're going to keep losing alleles because every individual is unique, right? So the existing diversity in this chemostat is going to drop rapidly as soon as we start the experiment. Eventually, given long enough time, there is only one fate of this population, that of all the alleles existing, n minus 1 alleles are going to get lost and only one allele will take over the population. Does that agree with everybody's intuition?
Yes, sir.
Yes, sir.
I have a question here.
Go
So,
ahead. Yeah.
assume that, for example, two or three mutations were. So, if n is pretty large, for example, in
Yes.
that case, maybe about 70. It's quite possible that maybe three or four of the mutations were already um, they multiply and become a huge proportion of the total number.
Yeah.
So it seems unlikely that now from these four populations, um, stochasticity would allow all of one particular population to go out,
Yeah.
right?
Yeah. Agreed.
So,
Yeah.
so in
So
that case,
yeah, go ahead. You just yeah, just complete what you're saying.
no. So in that case, I don't think that only one particular
Yeah.
mutation will survive.
Right, right. Absolutely. No, no, no question about that. So what Anirudh is saying, that if this population is indeed of the size 10 per 10 or something like that, then eventually agreed that the rate of loss of diversity is going to be very, very rapid early on. But the same stochasticity that we've been talking about so far is also going to ensure that when we reach equilibrium, we might have three different genotypes A, B, and C, and that they might constitute 20%, 30%, and 50% of the entire population. And in a scenario like that, and in a scenario like that, uh, 20% of a number as large as 10 per 10 is still 2 into 10 per 9. Then the thought that randomness is going to take place to an extent that I'm going to lose 2 into 10 power 9 individuals from this chemostat seems doesn't seem right, and hence as a result I could have coexistence of three different genotypes in the population. Does that does that summarize what you said, Anirudh,
Yeah,
accurately? Yeah, absolutely. So let me ask you this: are small populations more susceptible to randomness, or are large populations more susceptible to drift?
Small populations.
Small populations are much more susceptible to drift, right? If we started with our marbles in a jar game, let's say with 1,000 marbles, and if we started with 1,000 marbles, then obviously, and each of a different design, then we are going to lose marbles initially very, very rapidly, right? But then what's going to happen is that eventually we might get down to two different colors, red and blue, which are of the frequency 30% and 70%. And because 30% of 1,000 is still 300, getting rid of those 300 by drift is actually going to be extremely tough. Whereas if you were doing this experiment with 10 marbles, it's actually very easy to visualize in our head that loss of diversity or loss of designs in this case is going to be extremely rapid, and we could perhaps even actually play this game with 10 marbles and actually see all some of the 9 designs being lost in a setup like that. So the first is that small populations are much more susceptible to this, right? And we'll stop with this thought. So Anirudh, you're right that in large populations it doesn't work like that, right? Large populations, the action of drift is small. However, drift is a force which is relevant in small populations, and if you think of populations which are sort of uh, on the verge of extinction, numbers are small, and hence they become increasingly susceptible, not just because of selection, but also because of drift as a mechanism. The second idea is that there is something called a bottleneck effect. And bottleneck effect is something where you have a large population and you have some sort of an environmental catastrophe take place, and the population size suddenly shrinks. And when it shrinks, you only are left with a few individuals, or at the very least, most of the existing diversity that was there in the population is lost because of that reason. So you're only left with a much smaller population with a much smaller diversity in the population, and that's when drift sort of becomes a very relevant force. As far as bacterial populations are concerned or microbial populations are concerned, numbers are typically large. So drift wouldn't be seen unless in the case where environments are extremely sparsely populated, which is actually a fairly representative thing in an ecological setting where numbers are very small because of the presence of toxic chemicals, pathogens, predators. Numbers can be not very large. And also you have a couple of different forces acting on in addition to this. That one is that on top of this, you have bottlenecking effects, which we'll see. When we talk of how to do experiments in a lab, there are 3 ways to do it, and one of them is by introduction of bottlenecks, and we'll talk about that. And the other thing is that depending on the diversity that's left in the population, because of epistasis, the epistatic consequences of newer mutations occurring in this background are going to be different. If you have 1,000 different variants, the same mutation could have 1,000 different outcomes. But if you're only left with 3 different variants, you are only left with 3 different evolutionary trajectories. So genetic variation to that extent dictates, it gets coupled with selection and how that's going to impact populations and then determine the future evolutionary trajectories. So drift might not act in this direct way in a large population such as a chemostat in microbial species, but it has many different indirect ways of influencing populations. Okay. We will continue this last time over short time. So you can stay back if there are minor questions, otherwise we'll pick this up on Friday. Yeah, Pavitra, go ahead.
Sir, so in this chemostat kind of experiment, There is, so assuming that there will be some kind of scenario where there'll be only one mutation that survives after 3 or 4 days.
Yeah.
It comes because every other mutation also could have survived, right?
Yes. So if
So
you had 10 different chemostats whose starting point was exactly the same, after 3 days, all 10 would have a different mutation that survived.
okay, now after 3 4 days, if these cultures were like allowed to mate, it is possible that they would not be able to mate, right?
It is possible. Sure. Yes. But I mean,
Okay.
since we are talking bacteria, the sort of the question of mating doesn't arise here.
Okay, right.
Right. But you could think of this in yeast, for instance. Yeast can mate. So if you allowed one mating, yeast has two sexes. They're called A and alpha. If you allowed A type yeast in one and allowed it to evolve in one environment, if you allowed alpha type yeast to evolve in another environment, then there is a chance that after an extended period of time, they may not mate after that.
Right. Okay. Okay.
Yeah.
So that is why I said there could be different species in the beginning.
Yeah, I understand. Yeah. Sure. All right. If you are no more questions, feel free to move. Yeah. Sakshi, Yash, bolo.
Slightly off the topic, but I was thinking if number plays a large role in avoiding drift, then by that idea, individual species which live alone, not in a herd, those are highly susceptible to going extinct, right?
Yes.
But we don't see that happening that often, right?
Why do you say that?
I
I
mean,
mean, which species do you have in mind?
many predatory species tend to live alone.
Hmm.
For example, say crocodile. They don't live in a pack or a herd, right? But in a rainforest, we don't see a crocodile going extinct over, say, a few decades or so.
Yeah. Yeah. I mean, I don't know what are the kind of breeding values that a crocodile has. What are some of the selection pressures or something that a crocodile has? Good question. Let me think about that.
Okay.
Yeah.
I know it's off the topic, but
No, it's not off
maybe
the topic. It's not off
okay.
the topic. Yeah.
Okay.
Yeah. Sakshi, go ahead.
So I was asking, we are still not considering conjugation and other ways of reproduction in bacteria, right?
Yeah, we will not consider. So there are no. So there are no. If you're thinking of conjugation, transformation, transduction, we will not be considering how these modes influence evolution of these species, and there is a reason for that. That, at least for the purpose of this course, what I want to do is sort of not cover possibilities, uh, but sort of develop a theoretical framework in which we can look at evolution, right? So, like a concept like sequence space doesn't exist if we are looking at conjugation. If we are looking at transformation, that bacteria can just simply pick up pieces of DNA from the environment, the possibilities are just endless, right? You could pick up anything. Yeah? So there are no good theoretical ways in which we can envision those scenarios and lay down a firm conceptual framework in which to view processes such as this. Uh, so, for instance, one of the questions that we address in our lab is that how do genes cross species barrier? And that's a question of horizontal gene transfer, which could happen via the mechanisms that you're suggesting. For instance, that you pick up a gene from one species, you put that gene in another species, and chances are that it may or may not work. You don't know. But there is a good chance that if I don't know how many of you know codon bias and genetic code. Chances are that even that, so let me give you an example that we actually work on. We're going to take 5 minutes. Feel free to leave if you get bored or have something else. So one of the examples is that we pick up in yeast, every species has nitrogen transporters. They have ammonia transporters because nitrogen is essential. And then what happens is that what you could do, and people have done this, is that you can pick up nitrogen species. Yeast has its own nitrogen species. You can delete them, right? So now we have a species of yeast, variant of yeast, which if you grow in ammonia as the nitrogen source, it won't grow because all of its nitrogen transporters have been lost. We've deleted them, but this particular variant doesn't carry those nitrogen transporters. People have done this experiment for many, many decades. And what people have noticed is that if you take a human nitrogen ammonia transporter, put it back in this particular variant of yeast, the yeast does fine. So you took a human ammonia transporter, put it in yeast, and yeast goes fine. You took it from E. coli, you put it in yeast goes fine, right? And there are a couple of other species. C. elegans, you took a ammonia transporter, put it in yeast is fine. By fine, I mean it can grow in this ammonia in an environment where ammonia is the only nitrogen source. So there doesn't seem to be any problem, right? Genes are crossing species barrier, that you're picking genes from one to another, and they're doing fine. There's no issue with that. However, there is an amoeba called Dictyostelium, which is one of the classical model systems used, whose genome is very different from yeast. So genome is characterized by what percentage of your genome is GC. Guys, just give me 1 minute. Sorry. So in Dictyostelium's genome is very different from yeast, and because the reason that a genome is characterized, one of the ways to characterize a genome is that what percentage of the sequence is GC and what percentage is AT. A will always be equal to T. These are called Shargaf rules, which were discovered in the late 40s. So yeast has a certain GC percentage and Dictyostelium has a very different GC percentage. So there is a wide difference in the structure of these genomes. And what happens is that when we transport this nitrogen, when we pick up the nitrogen transporter from this amoeba and put it in yeast, it doesn't work, right? Although because genetic code is universal, this particular nitrogen transporter has to encode for the same amino acid sequence, but for reasons that we definitely can't go in now, this particular nitrogen transporter doesn't work. Whereas there are examples of at least 5 or 6 species where nitrogen transporters have been picked up and put in yeast, and they work fine. So the question now is that what enables a gene to cross this species barrier and be functional, and what are the scenarios where it cannot do that, such as the case in Dictyostelium, right? So again, I mean, I don't want to go into the details of the story. And then we try to create variants of this gene from amoeba, which would actually allow it to function in yeast. And then we try and understand that what were the fundamental changes that were taking place here. What sort of mutations did that gene need to acquire for it to be functional in a yeast genome, and whether this now edited version of the gene, which is functional in the yeast genome, functional in Dictyostelium, or has it lost its functionality in the Dictyostelium genome from where it came. So, I mean, we're interested in questions associated with how do genes move species barrier is the bigger question. The problem with this sort of stuff is that I couldn't tell you, nor am I aware of any resource which would be a good grounding way to study transitions of genes from one organism to another, uh, and allow us to understand in a theoretical framework which has some degree of universality associated with it. This part of understanding transitions or horizontal gene transfer events are sort of so few and so sporadic, and the evidence seems to be all over the place. There is, at least to the best of my knowledge, and a lot of people are doing a lot of work, so I'm definitely not aware of everything that's going on. But at least to the best of my knowledge, people are not aware of theoretical foundations which would allow us to study horizontal gene transfer events of the mechanisms that you mentioned. And it's for that reason that that's an area that I'm not going to touch. That one, there is a lack of theoretical foundations. If you gave me a nitrogen transporter from a species, I wouldn't be able to tell you whether this would function in yeast or not, or any other organism or not, if it were to uh, cross boundaries, or if it didn't work. I definitely, no one can tell you in the world that this is the number of mutations it needs to acquire for it to move from species A to B and be functional. So there is that lack of a theoretical framework in which to look at these events. And secondly, there is no end of possibilities as to what could get transferred. There are so much genetic diversity, so many genes, so many species. In an ecological niche, particularly, we'll be talking more about bacteria as we go forward, because these days most evolutionary experiments are done with bacteria and yeast because of the ease. That's the only reason. And in the ecological niche where these species live, yeast and bacteria, the amount of DNA that's present, the amount of other species that are present. I mean, one of my students in the group looks at how is genetic diversity maintained, how is species diversity maintained in an ecological niche. So we have scores of bacterial species isolated from soil samples on campus, and we look at how is this diversity maintained. Why is it that one is not taking over the population? What sort of relationships exist which allows all 20 species that we have isolated to remain together in this one grain of soil? So with that amount of species diversity in a population, it's very difficult to sort of understand that in a theoretical framework, and that's one of the reasons that we will not be talking about, definitely not be talking much about events such as this. I'm sorry, that was a long answer.
Thank you so much, sir, for the elaboration. But maybe, sir, this is the time that the theoretical foundation is being formed. Further, maybe the future generations will study.
Yeah, I mean, I think that's the goal of evolutionary biology, but I think we're still some distance away from that.
Thank
All
you,
right.
sir.
Yeah, sure. Let's close now. I have another meeting, so I will see you guys on Friday now. Okay?
Thank you, sir.
Yes, sir. Thank
Bye.
you,
Bye,
sir.
everybody.
Thank you, sir. Bye.
Bye.